This meeting is being recorded.
Party, my
This meeting is being recorded.
As a principal and a convener on behalf of my student, member of teaching faculty, non-teaching faculty, and myself, it is my privilege to welcome you all on online research methodology workshop on poster presentation competition. Dear friends, PSGP Mandal was established in 1969 under the kind and able leadership of Honorable Anna Saha PK Patil. Mandal has been serving the great cause of education in the remote Adivasi part of Maharashtra. This area is mainly populated with the economically and socially backward, poor people and landless Adivasis. To educate the young generation of such a poor section of society, Mandal started its Commerce and Science College in 1969, later on College of Engineering and Polytechnic in 1983. Then Industrial Training Center in 1985, College of Education in 1986, Management and Research Institute in 1987, the College of Pharmacy in 1994, Advanced Study Center for English and Indian Languages in 1995, College of Agriculture was started in 2003, Junior College of Education was started in 2004 and developed with all infrastructure and educational facility. At the present, the Mandal has owned continuous piece of 250 acres of the land. The Mandal honestly desired to offer the quality education in a young generation and make it competent enough to face the life in all difficulties, natural as well as the man-made. Now let me furnish some information about our College B Pharmacy course in Kuchhisane Gurti Vidya Prasarak Mandal was started in 1994. Under the kind and able leadership of late Honorable Annasa PK Patil, college was accredited by the NBA and it is approved by the All India Council for Technical Education and recognized by the Pharmacy Council of India. And it is affiliated to Kavitri Behnabai Chaudhary, North Maharashtra University, Jalgaon. 20 batches of young pharmacists have passed with the flying color. Many of them are serving in the pharmaceutical industries, institute in India as well as abroad. Our college is located in a peaceful and natural surrounding of Kuchhisane Vidya Prasarak Mandal campus. The college run in the 3-story building with additional basement by nearby all necessary facilities like voluminous library, well-equipped laboratories, classrooms, amenities for the sports and extracurricular activity. Students are encouraged for the preparation of various national level tests like GPAT, MBA, entrance, GRE, TOEFL, as well as the placement has been started in our college to provide a job opportunity to our students through the campus interview. Last year, our college conducted a campus interview and 89 students from all over the campus given the job. In every academic year, one national level seminar is conducted successfully organized by our college to provide the opportunity to our students to interact with the academicians and industrial persons. Well-known personality from the pharma sectors are invited as a speaker every year. Also, one day workshop on the personality development for the girls is successfully organized by our college under the heading Yuti Sabha, which is sponsored by the Student Development Section, North Maharashtra University, Jalgaon. I'm very proud to say that the Phase 1 Nandurbar District Avishkar 2018 was successfully organized by our college for the efforts taken for successfully organized. Even our college was appreciated by the university authority. We also highlight and award the local public for the social issue like cancer, AIDS, Swachh Bharat Abhiyan, acid attack, etc. by organizing the rally and street act every year. We are also adequate number of qualified teaching members who are competent, energetic. Their efforts mold our student into the well-informed, self-disciplined professional is appreciable. They also motivate the student should be qualify the competitive exam like GPAT for this purpose. We regularly organize the guest lecture. I would like to thankful the speaker for giving their valuable time for us, and would like to congratulate all organizing committee for coordinate for the successful arrangement of this event. I'm also thankful to Honorable Bill President Honorable Babasaheb Deepak Bhai Patil and Honorable Coordinator Amit Patil Sahib for always guidance and motivate us to do the best. Thank you very much. Thank you, Suresh sir.
Thank you, sir. I am very thankful to Dr. S.P. Pawar sir for introduction to our Mandal. Now I invite Dr. Sunila Patil ma'am, coordinator of this program, to introduce our guest and to talk with you. Please, ma'am.
Thank you, sir. Myself, Dr. Sunila Patil, as a coordinator of this event, on behalf of organizing team, I welcome all of you in the student development section, KBC North Maharashtra University Jalgaon sponsored one-day workshop on the research methodology and the poster presentation competition organized by PSGVP Mandal College of Pharmacy, Shahada. The event aimed to bring together different eminent personalities from the different field on one platform through this workshop on undelivered topics, and the poster presentation that will provide opportunity to interact with the academicians, scientists, and even enthusiastic our upcoming scientists, that is our students. The workshop, it comprises in the two sections. The section one is the guest lecture, which covers a range of topics on the research methodology, and section 2 is a poster presentation competition between the students from the different colleges. Now I introduce our eminent speaker, Dr. Nayan Gujrati sir. I'm very glad to say that sir is alumni of our college. His pass out batch is 2004. Sir is presently serving as assistant professor at SVKM Institute of Pharmacy, Dhure. Sir has guided total 24 MPharm students, then appointed as an expert for the Avishkar 2013 and 17 by KBC NMU Jalgaon. Sir has appointed as an NBA coordinator for the SVKM Institute of Pharmacy, Dhure. Successfully has completed various AICT, NITTT module, ARPIT, and MOOC courses. In his contribution as a researcher, he has published 14 international and 12 national papers in a well-reputed journal. Sir has published 1 reference book, 9 international chapters by the CRC Press, Taylor and Francis. He has delivered 8 expert talks as a resource person. Thank you, sir. Now I invite Dr. Nayan Gujrati, sir. Please, sir, take the charge of this session.
Thank you, ma'am. Thank you very much for such a nice introduction. Am I audible loud and clear?
Yes, sir.
1 minute.
Yes.
Yeah, 1 minute. I am going to share my screen. 1 minute.
Sir, now check it.
Okay, fine. Am I audible loud and clear to all?
Yes, sir.
Yes, sir.
Yes, sir. Yes, sir.
Thank you. Thank you very much. I am Dr. Nayan Gujarati, serving as Assistant Professor, Shri Vile Parle, Kelwani Mandal's Institute of Pharmacy, Dhule. First of all, I take this opportunity to thank Dr. Sunil Pawar sir for inviting me as a resource person for this workshop. I am specially extend my thanks to Dr. Suryanshi sir. He is my best friend, and Dr. Sunila Patel ma'am for such a nice introduction. She is my colleague. And I extend my thanks to all faculty members of Pujya Sane Guruji Vidya Prasarak Mandal's Institute of Pharmacy, as I started my professional career from this college only. I convey my warm greetings to all participants who joined this workshop. The title of my presentation today is Research Methodology. Dear learners, research methodology is itself a very broad topic. As a PG student and PhD scholar, it is one of the subjects which is introduced in the curriculum by university, and it requires a huge time period of say 6 months and a year to learn each and every parameter of research methodology. Today I will try my best to explain you some of the parameters that definitely count a lot for you when you actually involve yourself in research activity. So let's start our discussion by what do you mean by research itself? If you go through online resources and if you type research in Google search engine, the definition, there are a lot of definition you find out. As per my perception, research is a systematic investigation. I will call it as systematic because first of all, you have to go for a protocol. When you initiate your research activity, you have to prepare a plan of work, and systematically you have to proceed further in order to attain one next, say one by one, the step involved in your activity. In very simple term, I will explain you that research is a continual process. If you're on step first, let us consider one case study. Any person of your family is suffering from cough and cold. Then what you will do? You will identify some household remedy. You will go for any drug material or say any material which is present in your house, and you will start to apply to use that material in the treatment of particular situation. So what I am to say here that research is a continual process. When you are on step one, you have to only identify a slight change. You will identify a research, and based on that research problem, you will try your best, and based on literature survey, you will make some slight change in whatever the method in the treatment therapy available to you. Then you will enter from step 1 to step 2. If you observe step on, there is step 2. Now from step 2, we will move towards step 3 in same fashion. The step 3 is similar to step 2 with very slight modification. From step 3, you will enter on step 4. From step 4, you will enter on step 5. And when you will compare step 5 with step 1, it is completely different. So it is a step-by-step process. It is a continual process. whatever the situation which is exists in around to us, based on that situation, we have to do only slight modification with the help of literature survey. Here I have given one example, a very simple case study. It is applicable to all students of pharmacy as well as science. A person is suffering from XYZ infection, say cough and cold. Nowadays, it is a pandemic situation. So in an environment nearby to you, you find out that number of persons are suffering from cough and cold, and very simple household remedy available with us is use of ginger and tulsi. What exactly we are going to do? We will crush that ginger with the help of any, say, mortar and pestle, or with the help of the available resources in surround to you. You will extract active principle from that ginger and tulsi. Once you extract active principle, as a researcher, your next step is to go for purification of active principle with the help of chromatographic method and identify the activity of individual component of that extract. Once you find out, reveal the activity of individual component, you will come to know that in case of cough and cold, say particular principal constituent is said to be very essential, is found to be more beneficial, then next step is to convert that principal constituent into a product. And the product is available in market nowadays is in the form of cough syrup. So what we have done, the problem you have identified, and step by very slight modification with the help of literature, with the help of resources available in surrounding to you, we are going to perform activity one by one. And when we will reach to a final step, you find out that the step is completely different from first one, and that is called as research. It is a systematic investigation in order to establish facts to reach to new conclusion. What we are going to do, whatever the problem associated with society, that problem we have to identify, and we have to start our study. We have to study the material, we have to explore the resources, and once you explore it with the help of a proper protocol, then you will establish a fact and you will reach to a new conclusion. The new conclusion, as I belong to a field of pharmacy and my specialization is pharmaceutics, so normally I will look for a new formulation, a new drug delivery system, and that drug delivery system should overcome the drawbacks associated with conventional one. The conventional drug delivery system is available in market from last 1 decade, from last 2 decades. There are number of cough syrups which are available in market. There are number of steroidal formulation available in market. So what exactly I have to do? I have to identify the problem. I have to identify the drawbacks. And once I will identify the drawback, I will make suitable changes in order to establish the fact and reach to new conclusion. That is what called as research. Now, when as a student, I have to perform research activity. As a PhD scholar, I will look for any exact systematic protocol. First of all, I need a data. The next point is research data. With the help of available data, I will initiate any activity. So what do you mean by research data? Research data is nothing but information which is collected by you itself, observed or created for the purpose of analysis to produce original research result. The research activity is initiated by means of literature survey. There are a lot of workshops which are conducted on literature survey. How to do literature survey is said to be a crucial part of your entire journey. When I know that what is the method of literature survey? What are the genuine data available? It may be available through online resources or offline resources. We have to go through all official guidelines, which is related with that particular title. And if you know how to do literature survey, then only the researcher is able to identify the problem. So for that purpose, first of all, we will start our discussion with research data. The research data can be placed into two broad categories. The very first one is quantitative data and the second one is qualitative data. Now, quantitative data, it itself indicates that it is in the form of quantity. The quantitative data is used by the researcher to quantify a research problem. When I will identify a certain problem, I have to quantify it. If I wish to quantify that research problem, then such type of data which is available with me or the data which is collected by me through experimental activity is known as quantitative data. The quantitative data is gathered by means of instrument, with the help of instrument. I will give you a very simple example. In nanotechnology, it makes a revolution in the field of pharmacy as well as science. In nanotechnology, our first objective is to formulate a product which should lie in nano range. Now, in order to determine the size of particle, we have to use an instrument. And with the help of instrument, first of all, I will determine the size of each and every batch. And my first target is to prepare a formulation which should lie in nano range. Say your number of carriers for passive targeting in nanotechnology. In order to target your drug molecule to desired site of organ, the passive targeting is one of the method and active targeting is one of the method. In passive targeting, when you administer a drug molecule to any human being, the drug molecule will travel with the help of systemic circulation and through reticuloendothelial system, which is present in your body. The organs such as liver, spleen, heart will automatically get targeted. Now, how this organ or say why your drug delivery system will get accumulate to this organ, it is due to nano size. And in order to confirm nano size of your formulation, you have to use number of instruments such as zeta sizer, such as sonication, probe sonicators or ultrasonicators. With the help of that, you will make your product to lie in nano range and you have to determine the particle size of each and every formulation, and that type of data is known as quantitative data. Now, quantitative data is also said to be very beneficial from analysis point of view. Number of carriers nowadays available in order to deliver a drug molecule to different site of organ. Say cellular carriers are there, vesicular drug delivery systems, vesicular drug delivery system like liposome and neosome. The formulation is available in market as a novel drug delivery system. The formulation is available in pharmaceutical market. And this vesicular drug delivery system is categorized on the basis of their size. So on the basis of quantitative data. Say small unilamellar vesicle. Small unilamellar vesicle lie in the range of 1 to 100 nanometer. Then large unilamellar vesicle from 100 nanometer to 500 nanometer. And when you go for higher side, you find out that and even millimeter. So depend on the site to which you are looking for the drug should be targeted. You have to prepare your formulation and you have to adjust its size, its shape, its analytical parameter. And in that case, quantitative data will play a very crucial role. Now, quantitative data which should be counted. And in another, say for a student of the quality by means of quantitative data also. Let us consider a McDonald's cafe, and I have to judge the quality of food served by that cafe. What I will do, I will prepare a list of questionnaire, and that questionnaire, the answer of that questionnaire is on the basis of rating. The person used to visit the cafe will rate each and every question related to quality of food in the form of numeric scale on the rating of 1 to 4. So that type of data is also called as quantitative data, and quantitative data will also help you to reveal, to determine the quality. So there are two types. First, the data which should be counted itself, or the data that should be compared with the help of numeric scale 1 to 4. Now the second broad category of research data is qualitative data. When I will initiate my research activity as a student, the very crucial data is called as qualitative data, as it is going to describe the core characteristic. Based on that, the quality of your research project is revealed. What I want to say you that qualitative data is the data that will describe, that will characterize your whole project. And in order to collect a qualitative data, you have to perform an experiment. You have to perform an experiment in laboratory, as well as you have to record the observation. Now there are number of basic categories how you are going to collect the data. Based on method of collection, the data are discrete from each other. They are segregated from each other. Here you remember that the qualitative data in the field of science as well as pharmacy. I came to know from the host that the number of students who belongs to a field of science also, they joined this workshop. So I want to specially mention here that in case of science as well as life science, chemical science, physical science, or say pharmacy student, for that, we have to perform an experiment in laboratory and we have to record the observation. Now for the field of commerce, for the field of arts, they will go for a set of questionnaire. They will conduct interview, and with the help of interview, with the help of survey also, they are going to collect a qualitative data. And the next step is that the qualitative data is subjected for examination. Qualitative data, listen carefully all of you, qualitative data is always difficult to measure precisely. You have to come up with some new conclusion. You have to establish a fact. That is the basic goal of any research activity. For that, we have to measure the data precisely and we have to analyze it. And in order to analyze the data, nowadays, the number of quality management system tools are available. If you go through a recent activity in present scenario, the pharmaceutical industry are applying the quality management system tools such as quality by design, total quality management, Six Sigma approach, Six Sigma model have bring a revolution in the field of analysis. Once you get a quality data, if you subject it to Six Sigma model, then no doubt whatever the information you have revealed, whatever the conclusion you will going to make, will definitely contribute in real world process. It should be beneficial to society. For that, Six Sigma approach, Six Sigma model is utilized not only by pharmaceutical industry, but also by mechanical industry. Say Motorola have get a copyright for Six Sigma model in 2006, and they have reported a total profit of 600 million US dollar. What I'm saying, Motorola have reported the total profit of 600 million US dollar by application of Six Sigma model to their manufacturing division. What do you mean by this Six Sigma? I will explain you in brief about Six Sigma model, and I will assure you here that this Six Sigma model will help you to come up with a data that should be published in a journal with high repute. If you are looking for the publication in impact factor, if you want to complete your PG course, if you want to do your PhD, then nowadays every person asks you that where you have published your research data, what is your research outcome? And if you're looking for publication in a international journal, national journal with high repute, with Clarivate analytical impact factor, then you have to apply this type of quality management system tool. Say Six Sigma approach, say quality by design. I will explain you in brief about Six Sigma concept. This Six Sigma concept is specially applied for new methodology as well as existing process. If there is any existing process which is already approved by NDA, if I want to launch my drug product in pharmaceutical market, say any pharmaceutical industry, say Cipla, say Ocard, say Ranbaxy, they are going to market their product and they already take a permission of regulatory authority, say CDSCO. First of all, we have to take the permission of CDSCO and we have launched our drug product in market, and already the method is decided. Okay, and the method is followed by the industry. Now, if I feel that as a researcher, if I feel that I have to eliminate the defect which is associated with already approved method by NDA, with the help of NDA application, the method is already approved by regulatory agency and the product is available in market. Though I am feeling that I have to eliminate the defect, I have to improve the efficiency of manufacturing, then this model is appropriate. With the help of this model, we should eliminate the defect of existing model, and it is considered as a powerful tool that can transform a defect prone industry, a industry which is following a method consists of number of defects. So that defect prone industry is converted into an organization of perfection. They are known for, say, producing a perfect product with the help of with the help of process. So Six Sigma aims for performance target of only 3.4 defects. See the beauty of this method. It is aimed for performance target of only 3.4 defects behind every million activities, behind production of every million of products. Only 3.4 defects are known. So how it is applied in day-to-day research activity by a researcher, by a student of pharmacy, science and all? Very simple. There are two different models. The first one is DMAC model. It will stand for define, measure, analysis, improve and control. Define the problems. If you want to come up with any research activity in your laboratory, say as a student of first year M.Pharm, if you want to perform a research activity, it is your goal, or say assignment given by your teacher, what you will do? You should start with existing process, a manufacturing of tablet. A manufacturing of tablet, it is very well known to every student of pharmacy, say for science, say for life science student. You should start with any identification of microorganism in surrounding environment. So we are very well known with the procedure, how to perform it. Now what I will do, in an existing process, I will define all list. I will define each and every step involved in my process of production. Then I will enlist it. Simply, I will enlist all step. That is the very first phase to define. And what I will do, I will correlate each and every step with the quality of my end product. See, listen carefully. First of all, you have to define the problem, the research problem. You will enlist all different steps involved in your method. Which method? Existing method. I am saying existing method, which is available in your book, reference book, in literature. If you go through number of research paper, you find out that there is a well-known defined method has been established by number of research scholars. So same method I will note it. I will enlist all steps. And what I will do, I will correlate each and every step with the performance of my final product. How it is correlated? Which parameter of your final product is going to affect by this step? Second thing, the very second step is measure. In measure, what I will do, I will run the existing process, the current process. I will perform that process in my laboratory. And I will note down all quantitative data. I will gather all quantitative data. I will record observation with the help of instrument that will use to not only monitor, but collect the information. At the same time, whatever the qualitative data, whatever the descriptive data related to that process, I will note it. It comes under measure. Measure the ability of current process, how it is run successfully in your laboratory. Now in third case, that is analysis. Analyze. What you have to do, I am going to introduce you this model in brief at superficial level so that it is clear to each and every student who is going to attend this session. The third step is analyze. What do you mean by analyze? Very simple. Whatever the data we collected, that data is critically examined. Critical thinking, problem analysis is one of the major program outcome of every program. Program means a B.Pharm course, a BSc course, a BA course, BCom course. When I am performing research activity, this is the characteristic which I have to inculcate in each and every student. Problem analysis and critical thinking. Whatever the data I have obtained, that data I will thoroughly examine. How? What is my approach when I am going to analyze that data? I have to note down the undesired effect related to my data. I will go through, the data is already collected in measure phase. So I will go through each and every reading and I will note down what is the limit, official limit for assay given in IP. If the official limit of assay is given in IP is say 99% to 101%, what reading I have find out, and whether there is any undesired effect, say yes or no. If there is any undesired effect, that is called as analysis of data. And then once I have reported the undesired effect, I will try to improve the existing process. Because I have correlated already in defined phase, I have correlated the significance of every step with performance. So I came to know if there is problem with content uniformity will depend on mixing, and what is the process of mixing of my drug with excipient. If I know it, if I know the instrument, I will go for optimization. I will go for improvement of existing process. Once I will improve the existing process, then last step is to control, to correct the variation. To what extent should I deflect? To what extent should I change my process in order to get desired result? That is known as control. This is about first model. I will give you one example of pharmaceutical case study 2. The next model of Six Sigma approach is DMADV. It is the short form, abbreviation. D stands for define, M stands for measure, A once again stands for analysis. Then the next D here is to design. Now listen carefully. If as a researcher, I have to come up with new methodologies. Instead of existing process, I feel that the existing process is unable to meet the requirement of industry, meet the requirement of regulatory authority. If there is continuous failure in production batch, and now I have got an opportunity to design a new methodology, then this model will play a crucial. It will serve the purpose for me. Here, the fourth step is to design a new process, to come up with new methodologies that will completely eliminate, terminate the defect associated with your final formulation. And the last one is to verify the new process, verify the design methodologies in order to meet the official requirement. No doubt your product should be cost effective. No doubt your product should be efficient. If you go through FDA guideline, US FDA guideline, or say CDSCO guideline for new drug approval, the very first line given in that approval process is the drug should be effective and safe. What do you mean by effective? It should be effective as compared to available drug product in market. If for the treatment of fever, paracetamol is available in market, a tablet of paracetamol is available in market, which is very well known to every student here, present here, then I have to create a product which should be more effective than paracetamol. Then only my research proposal, my application will be considered by CDSCO. If I am coming up with new drug entity, that new drug entity should be more effective as compared to available drug product. And at the same time, we are playing with life. We are going to deliver our drug product to human being. So we should be utmost careful. And for that purpose, the safety consideration is said to be important, essential. And the line as per FDA is that whatever the new drug might be, whatever the new chemical entity you are going to launch should be effective and safe as compared to available drug in market. And the pharmaceutical industry nowadays are working on ANDA only, abbreviated new drug application only. Instead of NDA, new drug application, they are continuously striving lot on ANDA application. Whatever available drug product, you should pick up it from market. You should go for the same product with different process. So it is called as process patent. And with the help of that existing product in market by some different brands, we are going to file an ANDA application. We will create a same product and we will compare the bioavailability, the bioequivalence with a pioneer product. So that is what the pattern which is followed by pharmaceutical industry in India nowadays. And this model, Six Sigma model, will definitely help you to perform your research activity. This is the case study which is related to tablet manufacturing. For your kind information, why I am going to present it in front of you, because the same case study is applied by GlaxoSmithKline. Everyone is very well aware, very well known to GlaxoSmithKline. It is a popular brand. Number of drug products are available in market, and the product Zintac tablet is available in market in a dose strength of 150 milligram and 300 milligram. The ranitidine hydrochloride tablet. Maybe you already gone through that brand. Okay, so it is the case study which is associated with manufacturing of ranitidine hydrochloride tablet, and this case study is implemented Six Sigma model, application of Six Sigma model, DMAIC model by GlaxoSmithKline to the existing manufacturing process of ranitidine hydrochloride. What they have done? They have defined the steps involved in tablet manufacturing, say starting from weighing of raw material, blending, granulation, compression, and finally QC, quality control testing. Okay, it is further divided into three different categories: raw material testing, IPQC testing, and finished product evaluation. So what we are going to do exactly with the help of this model, Six Sigma model, DMAIC model, okay, you should successfully apply this small model in laboratory to various research title you are working with today. So I am going to explain you within a few minutes. Say in DMAIC, the first step is define. What they have done, they have enlisted the step and they have here enlist the critical point which is associated with that step that is going to impact your final product. The performance of final product is depend on this step by means of, say, blending. In case of blending, what is the type of blender you are going to use, how much is your time of blending. Blending means to mix your drug molecule with excipient. So no doubt the blending time will affect content uniformity, the blending yield will affect your assay, the type of blender is going to affect the whole process and the performance of your final product, because it is in case of unit doses from, say, tablet, exact dose of drug molecule should be delivered by single tablet. And when I am working in a batch of 10,000 tablet, 1 lakh tablet, I have to add the drug molecule in extent. Say I have to add drug molecule, the whole amount of drug molecule at a time. So how I should perform a blending and what should be its effect on final product, that is what the part of my first phase, define phase. Then what I will do in measure phase, I will perform the study. I will run the existing process. What I have already explained you, that in measure phase, I will run the existing process. And when I will run the existing process, what is my purpose? To collect the data, to collect a quantitative data. Say, first of all, I will prepare a granule. Why granules are prepared? To increase the flow property. Okay, you are very well known to it. What is the significance of granule? To increase flow property, to increase compressibility. So here, see, when I will collect the data related to granule, I will perform this parameter, bulk density. I will determine bulk density. I will determine tab density. I will determine Hausner's ratio. I will determine car price index, and I will describe it. I have to describe it to convert it into qualitative data. This is the quantitative data. Bulk density, tab density, the reading are quantitative data, and I have to describe it into qualitative, whether the flow property is excellent, whether it is good, whether it is flowable, whatever. Okay, at the same time, I will perform assay for n number of batches. See, listen carefully. In measure phase, you have to perform the activity on n number of batches, say at least 4 to 5 batch at laboratory level. At large scale level, you have to at least prepare more than 25 number of batches, more than 40 number of batches, and you have to collect the whole data. Then only you are able to reveal the defect, because my ultimate goal is to prevent a defect. That's why I have to run the process for n number of times, and I have to perform evaluation of granule, evaluation of the tablet to determine the ability of my existing process. That's why here I have determined each and every module. Here I have determined each and every characteristic related to granules as well as tablet. Once I got it, the next phase is analysis phase. In analysis phase, what you are going to do, you will identify the defect. Now, in order to identify the defect, whatever the data you obtain, you have collected a huge data, and that huge data is analyzed with the help of statistical tool. Biostatistics will also play a role. It will be a part of your research study, research activity. You have to apply statistical tools. Number of tools are available, Statistica and all. Say number of tools are there with the help of Excel, with the help of Statistica, with the help of modernized say applications. You are going to analyze your data, the whole data, and you will find out the undesirable effect. Say a study is performed by Dr. Swati Jagdale and associates for ranitidine hydrochloride tablet. This ranitidine hydrochloride tablet is prepared by GlaxoSmithKline and it is available in market in the name of Gintac tablet. So on same process of Gintac tablet manufacturing, the Swati Jagdale and her associates have applied this model, and when they go through the different batches, they have prepared 40 number of batches, and when they have collected data, they find out that there is variation takes place in the assay of ranitidine. The assay range is in between 146 to 152 for whole n number of batches. What I want to say you that I have run the existing process and I have performed assay to determine the concentration of drug in every batch. Randomly, I have collected the sample and I have subjected to assay procedure, which is already defined. So with the help of that procedure, I find out that the range of my drug molecule, ranitidine hydrochloride per tablet is 146 to 152 milligram. And the exact dose I want to deliver is only 150 milligram. So now there is variation in the range of assay for different batches. So I have to minimize this defect. This is said to be undesirable effect for me, and I will correlate this effect with the existing process, which is the factor responsible for this defect. So what I have done, I have gone for improve phase. In improve phase, I have to apply design of experiment DOE. This is very important tool, should be learned by each and every student belonging to pharmacy field as well as the field of science. If you are able to apply design of experiment, QBD, quality by design, then definitely whatever the existing process experiments, you are going to optimize it. And when I am going to optimize it, I will come to know the undesired effect and I will improve it, modify it. Now here, Dr. Swati Jagdale find out that the variation in assay is due to use of the blender. The existing blender is unable to mix your drug molecule with excipient. And then what she have done, she replaced the old blender with a new blender of higher capacity that will run at very high speed and able to mix uniformly the drug molecule through entire batch, and it will overcome the problem of assay. So in control phase, I have to perform the validation activity. Validation is also one of the crucial or say essential parameter of research activity. What do you mean by validation? It will be explained by your subject teacher. So in control phase, I have to go for process validation. Once I improve my process, once I replace the existing blender with new blender, once I define how the blending process should be carried out for my existing batch, then I have to perform process validation for 3 consecutive batches, and that is the part of control phase. So in this way, the DMAIC model is successfully implemented by GlaxoSmithKline with the help of Dr. Swati Jagdale and her associate, and she belongs to academic field. She act as a consultant for that industry, for that project work. They have performed it in industry, and they get a very good grant from the industry side, and they have successfully improved the production efficiency. And Glaxo itself reported the total benefit of more than 2000 US dollar due to application of this DMAIC model for production of ranitidine hydrochloride. So see, with the help of quality management system tool, quality by design, Six Sigma approach and all, you are able to collect the qualitative data. The qualitative data is in the form of descriptive word. It is in the form of narrative. It is available in narrative form, descriptive words, and that is subjected for examination in order to reveal the pattern or meaning. Are you with me, all of you? Are you with me, all of you? Yeah, I am clearly audible. Okay, fine, good. Thank you. So what I am saying, there is lot of disturbance. Please mute yourself. Yeah, there is some. Is there any question related to DMAIC model? Should I continue?
Yes, sir.
Should I continue? Okay, fine.
All the
Thank you.
participants
So
are
what?
requested, please mute yourself.
Yeah.
All
Yeah,
the participants
you,
are
Amit sir.
requested, please mute yourself.
So what I am saying, dear learners, that this DMAIC model or say qualitative data, when you gather that qualitative data, you subject it to examination and you have to reveal the patterns, or you should use coding. Coding will help researcher to categorize your qualitative data. Coding, nowadays coding also used to categorize your qualitative data, to identify the theme, and you have to address the research problem. First of all, you have to identify the problem, and then it is your duty, moral responsibility to address the research problem. And when you address the research problem, you are able to come up with new conclusion. At the same time, you will further subject it to quantitative analysis. And with the help of this different management system tool, you should able to publish your data in good journals, in quality journals, say with high impact factor. So the next one is method of collection. The collection of data is further divided into 2 different type. First one, the data which we have collected, say from different, say with the help of different methodologies, whatever we have collected data, it is further classified on 4 different types. First one is observational. Observational data is collected by means of observation of human behavior or research activity. Observation. This type of data is normally collected. A cohort study, an observational study, if you go through research methodology, you came to know that there are different types of studies. Say cohort study is there, observational study is there. Maybe it is not applicable for the field of pharmacy, but here we have to observe the behavior, the change in behavior of human. Say a particular category who born on this date, they should behave in different manner. There are a lot of studies, literature surveys are there, say survey on human beings, survey according to the date of birth, survey in response to any particular reaction when he gets scared, stage daring and all such type of behavior is noted with the help of observational data. We are interested in experimental data. As a student of pharmacy, as a student of science, we are always look for experimental data, and in experimental data there is an active intervention by researcher in an activity. Okay, the researcher involved in the activity by means of experimentation, by means of conducting, and he will prepare a list of questionnaire too. He will perform different tasks and he will collect the data by deflecting the variables. Listen carefully. When I am performing my research activity, I have to decide variable. What do you mean by variables? Variables are associated with your proposal. Okay, how to define your proposal, research proposal, that is different part. But when I am going to define my research proposal, I have to decide the variables. The variables are further divided into two categories, independent variable and dependent variable. And when I am going to variate my change my variables, I will perform the activity, research activity, experimentation, and I will come up with experimental data. So for experimental data too, you have to go for use of instruments. You have to define the methodologies and you have to draw a conclusion. Next one is the third category is derive or compile data. Derived itself, the word itself indicate derived means to derive means to create a data, qualitative data, right? I am talking about qualitative data, observational, experimental, followed by derived. Say a field of, for a field of commerce, for a field of your arts, such type of data will play a crucial role. Derived or compiled data, it is known as compiled data too. In case of derived data, what I will do, I will study the existing data points. I will study what? Existing data points. I will go for an extensive literature work available with me through different resources. See here, I will use different resources. And from different resources, I will collect the data and I will study the existing data point. And when I am going to correlate that existing data point from different resources with different thinking, perspective, and angle, I will create a new data, which is known as derived data. I will give you one example that will help you a lot to understand this concept. What do you mean by derived data? Very simple. Let us consider two metro cities. Okay, any, say Mumbai and Bangalore. These are the two broad metro cities in the map of our country. Now, when I will study these two different metro city from area point of view, I will get some data related to area of metro city. I will collect the data. This is my first angle. Then I will, second angle, I will collect the data related to number of population of these metro cities. So there are two different perspective, area of metro city and the number of population of that metro city. I will go for different resources online, offline, and I have collected all data related to these 2 points. And when I will correlate it, I will come up with new data that will give you the population density of metro cities. See, the third aspect which is generated population density with related to area. And then I will correlate it with different cities of our nation. So this is known as derived data. Maybe, I hope so, the concept is understand to all of you. Last one is simulation. Simulation is also one of the very interesting term. Simulation actually means initiation of situation or process. If you are able to imitate, to mimic the situation then you will definitely come up with a data which is required for real world process. It is all related to your real time activity. You have to simulate it, you have to mimic it. I will give you one very simple example with the help of computer model. Say a student of computer engineer, he come up with any software, he come up with a new application that will definitely help a lot to overcome the problem existing with real world. Then this type of data is known as simulation, the operation of real world process. Say I have developed a software that will help to engage the student and learn the concept. Gamification. Nowadays, when I go through the NIIT module courses, I came to know that there are number of application educational games are there. That educational games are in the form of software application which should be downloaded on your mobile. One of the very example, one of the live example of this educational game is Kahoot. Kahoot, this is an educational game which should be implemented by every instructor, every teacher in his class, that will engage the student to play a game. It is just like it will mimic the situation. It will create a situation that we are playing a game in a class, and that game, the educational game, will help to learn the concept. It will create a competitive environment. It will help the advanced learner to engage themselves in learning the concept as well as to perform better in competitive exam too. Kahoot, you should note it. So that is all called as simulation. Simulation means you have to perform a case study on existing world problem. That is known as simulation. So this is all about method of collection. On the basis of method of collection, the data is segregated, classified. Now next is research problem. When I have to initiate the activity, research activity in my laboratory, first very important thing is that I have to set an objective. What is the difference between aim and objective? We used to write down the aim. We used to write down the objective. What do you mean by aim? What do you mean by objective? The need of your research study, the rationale behind selection of drug molecule, the rationale behind selection of excipient material. If I want to prepare a mouth dissolving tablet, let us consider I have to prepare a mouth dissolving tablet. The tablet will get dissolved in my mouth. Which type of drug is suitable for mouth dissolving tablet? That what I have to identify. Listen carefully, all of you, dear students. When you want to come up with any activity, first of all, you have to set an objective, and an objective will run parallel to each and every experimental work you have performed in your laboratory. Your objective is step by step attained by different steps involved in your methods, and finally you will reach to conclusion. If the objective is not clear to you what exactly you have to come up with, then your whole activity will turns into zero. If I am going to prepare mouth dissolving tablet, for which drug molecule should I prepare mouth dissolving tablet? That is what my first question to me. I have to select, I have to give rationale behind selection of each and every material. Then for that, objective. There are two different type of research problem. First one is unconstrained. That is the problem on which there is no restriction at all. See here, when I am going to perform this practical in my laboratory, there is no restriction at all in the form of factors, in the form of variables. Then I will call it as unconstrained. Research problem. Research problems are divided into two categories. First one is unconstrained and second one is constrained. What is the difference between unconstrained and constrained? In unconstrained, there is no restriction at all. Say some instructor have asked me to prepare a tablet. that should be sufficiently hard. What is my objective? I have to prepare a tablet which is sufficiently hard. What you will do? You will go for application of compression pressure as possible, as much possible as you can. You will apply high compression pressure on that granules so that you will come up with a very hard tablet. There is no restriction at all in relate to compression pressure, in relate to hardness. Just you have to prepare a very hard tablet. That type of problem, research problem, is known as unconstrained. There is no restriction. But if I ask you the same thing, that you have to prepare a very hard tablet that should disintegrate within 15 minutes. The tablet, it is sufficiently hard. It should remain intact, but at the same time, it should disintegrate. When it comes in contact of gastric fluid, it should disintegrate, convert into particles within 2 minutes. Within how much? 2 minutes. Now there is restriction. When I am going to apply compression pressure, I have to also think about disintegration time. And disintegration time is said to be one of the factor which is going to reveal, which is going to produce impact on your final product. So constraint means what? There is restriction. When I am going to perform any activity, experimental activity, if there is any restriction related to levels, then it is known as constraint. And in that case, you should go for optimization. What you will do? You will go for optimization. The last one is variable. In case of variable, I will, just 1 minute. Okay. So what I am going to say, in case of variable, there are two different types. First one is independent variable and dependent variable. Variables are the factors, levels which are associated with your research activity, and when you are going to deflect it, you will get a qualitative data. So that topic is I will explain you because this is very important parameter. You are able to identify the variable, then definitely you will set a activity. You will come up with experimental activity. And this is my last point, and then we'll stop here. See, there are two different types of variable: independent variable and dependent variable. Independent variable means what? It is the factor that is associated with your process of research, process of experiment, and it is under the control of manufacturer, it is under the control of researcher. If I have to design any method, XYZ method, or I have to perform XYZ method, any method, it is either related to science student, say chemistry student, BSc chemistry student. If he want to synthesize a particular molecule in laboratory, then while synthesis of that molecule, he has complete freedom to select the raw material. Which type of raw materials, say what type of different ingredient I required for performance or for carrying out this synthetic activity in my laboratory. He have complete freedom to select material. He have complete freedom to add that material in defined quantity as desired by him. So these are the factors which are under control of researcher. If I belongs to a pharma background, if I am performing any simple experiment in my laboratory, then there is complete freedom for certain variables, certain parameters that I will select by own. And that type of variables, factors are known as independent variable. Now this independent, you have to prepare a list of independent variables. See, listen carefully. I will insist you all that when you are performing any activity, you have to first of all list out the independent variable. Once you list out independent variable, you have to go for determination of relationship. In qualitative data will allow researcher to develop a causal relationship between independent variable and dependent variable. What do you mean by dependent variable? The second category here is dependent variable. Dependent variable are nothing but responses. These are the responses. When you will go for deflection, when you will go for change of any particular factor that will impact on the characteristic of your final product, and which characteristic is going to affect, that is known as dependent variable. If I am going to alter the mixing time of drug molecule with excipient, it is going to affect my drug content uniformity. If I am going to increase the temperature during synthesis, it is going to affect the degradation of my drug molecule or excipients. So that is all dependent variable responses. And these responses, you have to find out the relationship between responses and independent variable. And here your actual research lies. If you are able to find out the relationship between the factors and responses, then you are able to optimize it. And when you are able to optimize it, you will come up with new product. You will come up with new conclusion. When I have to address the research problem, first of all, what I will do, I will identify the problem. I will give an objective that I have to find out the remedy for cough and cold. Now I will, as a pharmaceutical, as a as a student of pharmacy background, or say for pharmaceutical research, I have to study the etiology of diseases. That is a different part. As a science student, if you want to synthesize, if you want to increase the yield of synthesis, then instead of reflux, you will go for microwave. When you will go for microwave, when you came to know that I have to increase the yield, I have to maintain the temperature, I have to control the reaction, then these are the factors which are under the control of manufacturer, researcher, and the responses. When that factor is deflected, what is the response on your ultimate process? I think so I am able to differentiate independent variable and dependent variable. Next one is factorial design. In case of factorial design, you have to identify factors as well as label. And the last one is application of design of experiment 2s to 2 as well as 3s to 2. It is say pronouns abbreviated as 2s to 2. Never called it as 2 cube. It is 2s to 3 or say 3s to 2. What this 2 indicate here? The 2s to 3. In this 2 indicates the label and 3 indicates factor. In 3s to 2, listen carefully, all of you. Try to understand 3s to 2. In 3s to 2, 3 indicate the label and 2 indicate factors. How many number of factors? Factors means what? Independent variable. Factors means what? Independent variable. The parameter which are under the control of researcher. And label means what? +1, 0, -1. These are the 3 different labels which you have to take in order to apply design of experiment. If I am going for 3s to 2, I have to prepare how many number of formulation? 9 different formulation. 3s to 2 means 3 into 3. 3 into 3, it becomes 9. So 9 different formulation I will prepare in my laboratory with different formula. All these 9 formulation have different formula. I will explain it if the time permits me. Actually, we are running too late. It is already 12 o'clock. So I think so this much is sufficient. When I am going for 3s to 2 design of experiment, it indicates 3 into 3. That means 9 different formulation, 9 different batch, 9 different type of experiment practicals I have to perform in my laboratory, and all these experiment will vary from each other. And whatever the responses for all these 9 experiment, I will record. And based on that qualitative data, I will decide which batch is said to be an optimum one, optimized one. Okay, so for optimization, it is very essential. Research guidelines are there. In research guidelines, a research methodology is itself help you to prepare your whole protocol and your come up with a thesis includes title, how you have to define the title of any research work. The title itself, please please mute yourself. I request host to unmute to mute all. Yes. It is a last slide. Please concentrate. With the help of research guideline, see there are different
Questions करने दो पहले.
headings, title, rational, drug profile, material and method, result and discussion, followed by conclusion and finally future prospects. So these are the 8 parameters which you have to cover up with your research activity. Each and every heading is itself a very broad topic. If I am going to give a title for my research work, what should be the approach of researcher? The title should be self-explanatory. In 2013, I have completed my PhD, and one of the very important conditions laid down by my research guide is to publish the research work in international journal with high repute, say impact factor. Then I have working on microsphere, and microsphere is said to be a very general topic in 2012-13. Already a lot of work had been published. So what I have done, I have come up with the same research topic, and in that research topic, I have designed the title of my research topic in innovative way. The title is going to play a very significant role. What I have done, I have prepared microsphere only, but I have prepared that microsphere with the help of 2 different polymer. So I have redesigned my title. Instead of microsphere, I have written down polyelectrolyte complex, because there are 2 different polymer which I am going to use, which will act as a carrier for my drug molecule. And that 2 polymers are going to form a complex with each other. The first polymer is going to form a complex with second polymer. And in that complex, the drug molecule is entrapped. So what I have done, I have written polyelectrolyte complex in my title. Followed by that, my complex possesses a characteristic of pH sensitivity. When it enter into stomach, it will get constrict, and as soon as it reach to intestine, it will swell and the drug will be released. So what I have done, I have written pH sensitive polyelectrolyte complex, and that refine my entire project work. So with the help of title, I have given an innovative touch to my existing research work. It is, though I have formulated microsphere, but I incorporate the mechanism of that microsphere, how it is working in your body with the help of title itself. The title should be self-explanatory. You should use different quality management system tool in your title, and that will make your title, that will give a research touch, that will give a innovative touch to your title, and that title will be published. So each and every heading will play a very important role when you are looking for publication, when you are looking for, say, higher impact factor journals, and if you have to go for competitive competition, different research competition, you should apply your own knowledge. So I think so this much information is sufficient for today. I will complete my presentation here, and I will really interested if there is any question from your side, I will address it. Thank you. Thank you very much.
If anyone have any question, you can ask. Students can ask the question to
Thank you.
answer.
Yes.
Okay. I would like to invite Amit Dangani sir for concluding this session. Amit sir.
If there is any question from any participant, I am really happy to address it. If there is any one of you. Yes. Okay, fine. Thank you. Thank you very much. Thank you, Dr. Sunil Pawar, sir, Principal of College of Pharmacy, for inviting me to deliver this talk. I am really feel so proud when I am in front of all audience of PSGP College of Pharmacy Shahada, because it feels me great. I have completed my B.Pharm from this college. I have started my professional career, and really there is a different touch associated with this college. So I am really thankful to Principal Sir for giving me this opportunity, and I am also thankful to each and every faculty member. They are very good friend to me. They are always being supporter for me in different situation. Dr. Hemant Suryavanshi sir, Dr. Jayesh Patel sir, Dr. Say Professor Hamid sir, Kadvi sir, and all faculty members. I really appreciate. Thank you very much, sir. Thank you, sir. Thank you, Pawar sir. I hope so. I meet your expectation.
Thank you, sir. Thank you very much. It is indeed always a pleasure to hear you, sir. Not only this session, but right from my college days. Various concepts of research methodology like Six Sigma concept, DMAIC model, DMADV model, and their application in science and pharmacy was very simply and easily explained by Dr. Nayan Gujrati. Also, these concepts were justified very keenly with the help of simple and day-to-day examples. Sir, your session will really help our students and participants in designing their research very effectively. Once again, thank you very much, Dr. Nayan Gujrati, for delivering such an informative session. Thank you, sir. Thank you very much.
Thank you very much. Should I leave now? Yeah. Thank you very much. Should I leave? Yes.
Yes, sir.
Okay.
Okay, thank you, Nayan sir.
Okay. Bye bye. Thank you, sir. Thank you very much.
Thank you.
Bye bye. Yes. Bye bye, sir. Have a nice day, sir.
Okay, thank you.
Over to you, Dr. Hemant Suryavanshi.
Sir, please introduce our second guest, sir. Amit, sir.
Okay. Our second resource person for today is Dr. Tushar Patel. Currently, he is working as head Department of Marathi, Pujya Sane Guruji Vidya Prasarak Mandal's Art, Science and Commerce College, Shahada. He has completed his PhD in the year 2016 from Pune University, Pune. He has 16 years of teaching experience. He has published several books on Gurjari Sahitya. He has also published various research papers on various subjects. He is also designated as Joint Secretary, Alumni Association of Pujya Sane Guruji Vidya Prasarak Mandal's Art, Science and Commerce College, Shahada. I would like to invite Dr. Tushar Patel for the session. Over to you, Dr. Tushar Patel, sir.
हॅलो.
Yes,
नमस्कार. येतोय आवाज?
Yes.
ओके. नमस्कार. आजच्या या रिसर्च मेथडॉलॉजी संशोधन पद्धतच्या कार्यशाळेमध्ये मला संयोजकांनी बोलण्याची संधी दिली, त्याबद्दल मी संयोजकांचा आभारी आहे. संशोधन पद्धत. आता संशोधन म्हटलं की रोजचं आपण जे काही आयुष्य जगत असतो, त्या आयुष्य जगत असताना वेगवेगळ्या समस्यांना त्या ठिकाणी आपल्याला सामोरं जावं लागत असतं. गरज ही शोधाची जननी आहे, हे वाक्य आपण नेहमी ऐकत असतो. आणि अतिशय जुन्या काळापासून मानवासमोर निसर्गाने जी काही परिस्थिती निर्माण केलेली आहे, त्या परिस्थितीची झगडत असताना, त्या परिस्थितीशी आपण तोंड देत असताना वेगवेगळ्या समस्यांना आपल्याला तोंड द्यावं लागत असतं. त्या समस्यांना लढून आपण त्या ठिकाणी आयुष्य जगण्याचा त्या ठिकाणी प्रयत्न करत असतो. त्या समस्या सोडत असताना ज्या वेगवेगळ्या अडचणी आपल्याला त्या ठिकाणी येत असतात, त्या अडचणी सोडत असताना जी धडपड आपण करतो, त्या धडपडातूनच काहीतरी नवीन त्या ठिकाणी निर्माण होत असतं आणि त्याला त्या ठिकाणी संशोधनाचा कुठेतरी प्रारंभ किंवा संशोधनाची कुठेतरी सुरुवात असं आपण त्या ठिकाणी सांगू शकतो. एक रिसर्च म्हणजे काय की नवीन काहीतरी गोष्टीचा एखादा शोध घेणे आणि त्या गरजेतूनच त्या शोधाची त्या ठिकाणी निर्मिती ही होत असते. संशोधन म्हणजेच काय की एक वैज्ञानिक कार्यपद्धती जी काय असते ती वैज्ञानिक रीत आपण जे सरांनी जे सांगितलं मगाशी की वेगवेगळ्या टप्प्यांच्या माध्यमाने वेगवेगळ्या पद्धतीच्या माध्यमाने जे काही आपण एखादी समस्या जी काय सोडत असतो ते सुव्यवस्थितपणे आणि नियमबद्ध अभ्यासक्रमाची जी काही प्रक्रिया आहे त्याला त्या ठिकाणी आपण संशोधन असं म्हणत असतो. संशोधनाच्या त्या सगळ्या क्षेत्रामध्ये वेगवेगळ्या त्या ठिकाणी व्याख्या आपण अभ्यासत असतो. वेगवेगळ्या व्याख्या त्या ठिकाणी केल्या गेलेल्या आहेत आणि त्या सगळ्या या व्याख्यांचा जर आपण संदर्भ बघितला तर एखादी गोष्ट शोधून काढण्यासाठी सतत आणि पद्धतशीरपणे जे काही परिश्रम त्या ठिकाणी विद्यार्थी करत असतो त्याला त्या ठिकाणी संशोधन असं म्हटलं जातं. ज्ञानाचा जो काही विस्तार आहे तो ज्ञानाचा विस्तार, सुधारणा, पडताळा या बाबतीमध्ये सामान्यीकरण करण्याच्या हेतूने गोष्टी, संकल्पना व प्रतीके याची जी काही मांडणी त्या ठिकाणी आपण करत असतो तेच म्हणजेच काय संशोधन होय. थोडक्यात म्हणजेच काय की संशोध घेण्याच्या उद्देशाने चिकित्सकपणे सतत आणि पद्धतशीरपणे केलेले परिश्रम याला त्या ठिकाणी संशोधन असं म्हटलं जातं. अज्ञात असणाऱ्या ज्या आपल्याला माहिती नाहीयेत अशा ज्या काही गोष्टी आहेत त्या शोधायला जिज्ञासा ही कारणीभूत ठरत असते. जाणून घेण्याची जी काही माणसाच्या वृत्तीमध्ये जी काही एक स्वभाव वृत्ती जी काय असते की एखादी गोष्ट जी काही आहे ती जाणून घ्यायची आहे, माहिती करून घ्यायची आहे. तर अशा ज्या काही अज्ञात ज्या काही गोष्टी आहेत त्या अज्ञात असणाऱ्या ज्या काही गोष्टी शोधून किंवा त्याची माहिती काढून जाणून घेण्याची जी काही संशोधकामध्ये वृत्ती असते त्या वृत्तीला किंवा त्याच गोष्टीला संशोधनामध्ये सर्वात वरती प्राधान्य हे त्या ठिकाणी असतं आणि त्यासाठी जी काही आहे वेगवेगळ्या पद्धतीने कृती केली जाते. जसं आपण एखादा प्रयोग ज्या वेळेला करतो प्रयोगशाळेमध्ये तर त्या प्रयोगशाळेमध्ये एक टप्प्याटप्प्याने आपण त्या प्रयोगाची मांडणी केलेली काय असते त्या टप्प्याटप्प्याने तो प्रयोग त्या ठिकाणी करत जातो आणि शेवटी निष्कर्षापर्यंत आपण पोहोचत असतो की तुम्ही काय सिद्ध केलं. तो जो काही प्रयोग तुम्ही केला असेल एखादा रसायन दुसऱ्या रसायनामध्ये ज्या वेळेला मिश्रण होत असतं त्याच्यातून तिसरं काहीतरी एक निर्माण होत असतं. त्यासाठी विशिष्ट अशी प्रक्रिया पार पाडत असते. त्याला आपण 1 नाही 2 नाही तर 10 वेळेला पडताळा करून बघत असतो आणि मग शेवटी आपण जे काही त्या ठिकाणी निष्कर्ष त्या निष्कर्षापर्यंत पोहोचतो की आपण ती प्रक्रिया केल्यानंतर नवीन काहीतरी त्याच्यातून आपल्याला हा शोध लागला. अगदी तशाच प्रकारे या साहित्याच्या क्षेत्रामध्ये कलेच्या क्षेत्रामध्ये त्या संशोधनाची एक पद्धत आहे की त्या ठिकाणी आपण एखादी गोष्ट डोळ्यासमोर ठेवतो, एखादा विषय डोळ्यासमोर मांडतो की या विषयावर संशोधन ज्या वेळेला आपण करायचं ठरवतो की संशोधन करण्यासाठी मग सर्वप्रथम त्या ठिकाणी सत्य त्याच्यातलं काय आहे हे शोधणं त्या संशोधकाचा त्या ठिकाणी प्रमुख हेतू असतो. एखादी गोष्ट एखादा विषय मांडल्यानंतर त्याच्यातला जे काही सत्य जे काय असेल जे काही वास्तव असेल ते शोधणं ते मांडणं हे त्या संशोधकाचं प्रमुख काम असतं, मुख्य काम असतं आणि संशोधन ही न संपणारी अखंड अशी प्रक्रिया आहे. संशोधन ही प्रक्रिया कधीच थांबत नसते. समस्या ज्या वेळेला दूर होते त्या संशोधनाच्या माध्यमाने ज्या वेळेला आपण शोध घेत असतो त्या शोधच्या माध्यमाने ज्या वेळेला ती जी काही समस्या असेल ज्या समस्येचे निराकरण करण्यासाठी आपण प्रयत्न करत आहोत जी समस्या आपल्याला निर्माण झालेली ती समस्या सोडवण्यासाठी आपण संशोधन करत आहोत तर ती समस्या दूर करून सतत नवीन ज्ञानाचा शोध त्या ठिकाणी घेत राहणं, ज्ञानात भर घालत राहणं हे त्या ठिकाणी त्या संशोधनामागची प्रमुख भूमिका असते. आणि मानवी जीवनाला संशोधन नेहमी शुद्ध करत असतं, नवीन नवीन ज्ञानाचा त्या ठिकाणी भर घालत असतं. संशोधन म्हणजेच काय की ज्ञानाच्या त्या ठिकाणी शोध घेणं. आपण मगाशी बघितलं कोणतीही जी काही समस्या असते ती शास्त्रीय पद्धतीने कशी सोडवायची हे त्या संशोधनातून आपल्याला त्या ठिकाणी कळत असतं, आपल्याला त्या ठिकाणी माहिती असतं. एक विशिष्ट असे टप्पे त्या ठिकाणी आहेत. मग त्यामध्ये मग तुम्ही संशोधनाचा प्रामुख्याने पहिल्यांदा हेतू काय आहे तुमच्या संशोधनाचा, तुम्ही तो विषय का निवडला, एखादा विषय जर निवडला असेल तर त्या विषयाचा तुम्ही निवड का केली, इथून अशी सुरुवात होत असते. मग त्याच्यामध्ये तुमचा संशोधनाचा हेतू तर मग त्याच्यामध्ये तुम्ही हेतू काय म्हणून ते ठेवलेला आहे त्याची मांडणी त्या ठिकाणी करावी लागते. त्याच्यानंतर उद्दिष्टे, की त्या ठिकाणी संशोधन करत असताना, ते रिसर्च करत असताना तुम्ही कोणकोणती उद्दिष्ट डोळ्यासमोर ठेवलेली आहे, त्या संशोधनातून सर्वात शेवटी तुम्ही काय मांडणार आहात जनतेसमोर, किंवा त्या ज्ञानामध्ये किंवा त्या अभ्यासामध्ये त्या विषयामध्ये तुम्ही काय भर घालणार आहात, तर ते उद्दिष्टांची मांडणी त्या ठिकाणी करावी लागते. त्यासाठी मग साहित्य कोणतं वापरणार आहात, संदर्भ ग्रंथांची जी काही सूची असेल, सपोज तुम्ही जे काही संदर्भ ग्रंथ जे काही तुम्ही त्या संशोधनासाठी अभ्यासासाठी वापराल, त्या संदर्भ ग्रंथांची सूची त्या ठिकाणी तुम्ही त्या संशोधनाच्या त्या आराखड्यामध्ये तुम्हाला जोडावी लागते. आणि अशा काहीतरी संशोधनाची कृती, असे जे काही टप्पे आहेत, अशी जी काही प्रक्रिया आहे, या सगळ्या पद्धतीने परिपूर्ण असं ते संशोधन जे काही असतं त्याला शास्त्रशुद्ध संशोधन असं त्या ठिकाणी संबोधलं जातं, म्हटलं जातं. ज्ञानाच्या पलीकडे जाऊन सत्य तपासून पाहण्यासाठी आपल्या जी काही माहिती आहे, आपल्याला जी काही कल्पना आहे, त्याच्या पलीकडे जाऊन जे काही वास्तव आहे त्या विषयामागचं जी काही सत्यता आहे, ती सत्यता तपासून पाहण्यासाठी किंवा माहिती मिळवण्यासाठी संशोधन हे आपल्याला त्या ठिकाणी मदत करत असतं, की जे मांडून जे सगळं आपण अभ्यासपूर्ण मांडणी करून आपण त्या ठिकाणी त्या विषयामध्ये नवीन काहीतरी भर घालत असतो, नवीन मांडणी त्या करत असतो, किंवा समोरच्या साहित्यामध्ये काहीतरी एक नवीन आपण त्या ठिकाणी भर त्या ठिकाणी घालत असतो. संशोधन म्हणजे एक शास्त्र आहे, ती एक कला आहे, आणि त्या शास्त्राच्या त्या ठिकाणी मगाशी सांगितल्याप्रमाणे पायऱ्या आहेत. त्या पायऱ्यांनी जर तुम्ही जर संशोधनाची प्रक्रिया पार पडली तरच ते संशोधन तुमच्या शास्त्रशुद्ध पद्धतीने त्या ठिकाणी परिपूर्ण होतं. जसं आपण गणितामध्ये बघतो की गणिताच्या काही विशिष्ट अशा पायऱ्या असतात, सूत्र असतं, ते सूत्र ते तुम्ही व्यवस्थित जर मांडणी जर केली, त्या सूत्रांच्या माध्यमाने ज्या काही पायऱ्या असतील त्या गणित सोडवण्याचा, त्या पायऱ्यांच्या माध्यमाने जर तुम्ही टप्प्याटप्प्याने जर ते गणित सोडवत गेलात तर तुमचं शेवटी उत्तर हे त्या ठिकाणी बरोबर येत असतं, किंवा त्या गणिताची त्या ठिकाणी सोडवणूक ही व्यवस्थितपणे होत असते. तशाच प्रकारे संशोधनामध्ये देखील त्या पायऱ्या ज्या काही आहेत, परिकल्पनेची मांडणी असेल, परिकल्पनेच्या ध्वन्वार्थ असेल, माहितीची संकलन असेल, विश्लेषण असेल, जी काही माहिती तुम्हाला प्राथमिक मिळालेली आहे त्या विषयासंदर्भात, त्या माहितीचे संकलन तुम्ही कशा पद्धतीने केलेलं आहे, ते माहितीचे संकलन योग्य झालेलं आहे का नाही, त्याचे विश्लेषण तुम्ही कशा पद्धतीने केलेलं आहे, त्याच्यानंतर मग जे काही तुम्ही परिकल्पनाचा पडताळा असेल, त्या सगळ्या ज्या काही पायऱ्या आहेत त्या संशोधनाच्या, हे जे काही टप्पे आहेत त्या संशोधनाचे, या सगळ्यांच्या आधारे एखादं जे काही संशोधन तुम्ही त्या ठिकाणी करत असतात, जो विषय तुम्ही हाताळत आहेत, त्या संशोधनाला त्या ठिकाणी या टप्प्यांच्या माध्यमाने दिशा देता येते. एखादं संशोधन कार्याला ज्या वेळेला आपण सुरुवात करतो, संशोधन करण्यापूर्वी त्याच्या संदर्भातली जी काही पुस्तकं असतील, मासिकं असेल, संशोधन अहवाल असतील, याचं वाचन त्या ठिकाणी त्या संशोधकाने करणं आवश्यक असतं. त्याची प्राथमिक जी काही माहिती आहे ती माहिती त्या संशोधकाला असणे आवश्यक असते, कारण या आधारावर संशोधक हा संशोधनासाठी विषय त्या ठिकाणी निश्चित करू शकतो. कारण नुसतं संशोधन करायचं म्हटलं तर काय करणार, त्यासाठी तुम्हाला प्राथमिक काहीतरी विषय तरी तुमचा त्या नजरेसमोर असावा, आणि तो विषय तुम्ही कधी सुचवता, किंवा तुम्हाला तो कधी विषय सुचेल की ज्या वेळेला तुम्हाला त्या विषयाचं काहीतरी प्राथमिक ज्ञान आहे, किंवा मनातून तुम्हाला त्याची आवड आहे, आणि ते ठिकाणी वाचन करत असताना, त्या ठिकाणी तुम्ही त्या विषयाचा धांडोळा घेत असताना तुम्हाला काहीतरी समस्या येतात, किंवा तुम्हाला काहीतरी जाणीव होते की याच्यातून काहीतरी असं निघू शकतं बा, किंवा असं या विषयाची मांडणी होऊ शकते. तर अशा वेळेला ते प्राथमिक जे काही वाचन आहे, त्या वाचनाच्या आधारे ज्यावेळेला आपण ते मांडणी करत असतो कल्पनांच्या माध्यमाने, त्यावेळेला आपल्याला ती गरज भासते, आणि त्या गरजेतूनच त्या ठिकाणी त्या नवीन गोष्टीचा शोध घेण्याची जी काही एक मानसिक इच्छा जी काय आहे, जिज्ञासा जी काय आहे, ती त्या ठिकाणी निर्माण होत असते. आणि अशाच माध्यमाने त्या ठिकाणी संशोधनासाठी त्या ठिकाणी तो विद्यार्थी किंवा तो संशोधक विषयाची निश्चिती करू शकतो. आणि एक वेळेस विषय मग तुमचा जर निश्चित झाला की मग संशोधनाचा आराखडा त्या ठिकाणी तुम्हाला तयार करायला हे अतिशय सोपं जात असतं. कारण साहजिकच आहे की ज्या अगोदर आपण कुठेही तर आपण एखाद्या कामाला सुरुवात करतो तर त्याची एक प्रारूप आराखडा जो काय असतो, प्रारूप रचना जी काय असते ती आपण आपल्या डोक्यासमोर ठेवतो. एखाद्या प्रवासाला जर आपण निघालो तर आपण तो प्रवासाचा एक दिशा ठरवतो की बा इथून असं जायचंय, त्या ठिकाणी थांबायचंय, तिथून तसं जायचंय. अशा हिशोबाने आपण म्हणजे पूर्वनियोजित आपण जे म्हणतो, पूर्वनियोजन जर करून आपण जर गेलो तर त्या ठिकाणी ते काम सुखरूप पार पडत असतं किंवा व्यवस्थित त्या ठिकाणी पार पडत असतं. तशा प्रकारे जर त्या संशोधनाचा ढोबळ विषय जो तुमच्या डोक्यात जर राहिला वाचनाच्या आधारे, तुमच्या प्राथमिक अभ्यासाच्या आधारे, तर त्या ठिकाणी तुम्हाला मग त्या ठिकाणी तो विषय निश्चित झाल्यानंतर संशोधनाचा जो काही आराखडा आहे तो आराखडा तयार करायला त्या ठिकाणी सोपं होतं. आणि एक वेळ संशोधनाचा आराखडा जर तुमच्या समोर तयार झाला तर त्या ठिकाणी मग ते संशोधन जे काही आहे, जे तुम्ही संशोधन करणार आहात, जो तुम्ही अभ्यास करणार आहात, तो टप्प्याटप्प्याने मांडणी करायला देखील तुम्हाला त्या ठिकाणी सोयीस्कर होत असतं. संशोधन आराखडा म्हणजेच काय, की एक प्रकारे ज्या विषयाचे तुम्ही संशोधन करणार आहात त्या संशोधनाची एकंदरीत तुम्हाला तो दिशा दाखवतो तो आराखडा. तुम्हाला कशा पद्धतीने जायचं आहे शेवटच्या निष्कर्षापर्यंत, शेवटच्या संदर्भ ग्रंथांची जी काही आपण सूची जोडतो त्या संदर्भ ग्रंथाच्या सूचीपर्यंत तुम्हाला ते एक तो आराखडा जो काही आहे तो दिशा दाखवण्याचं काम करत असतो. मग त्या आराखड्यामध्ये तुम्ही अभ्यास कोणत्या केंद्रावर, म्हणजे अभ्यास केंद्र कोणतं आहे, तुमचा अभ्यास कुठं करणार आहात, संशोधन अभ्यास पूर्ण होण्यासाठी त्या ठिकाणी किती कालावधी लागणार आहे, तुम्ही कोणत्या पद्धतीचा अवलंब करणार आहात, माहिती संकलनासाठी तुम्ही कोणती साधनं वापरणार आहात, अशा प्रकारे पूर्ण गोष्टींचा त्या ठिकाणी संदर्भ, पूर्ण गोष्टींचा उल्लेख त्या आराखड्यामध्ये त्या ठिकाणी येतो. मग त्याची गृहीतक काय, मग अशी सांगितल्याप्रमाणे उद्दिष्टे काय, त्याची व्याप्ती काय, संशोधनाच्या मर्यादा काय, कारण विषय वेगवेगळे असतात त्यानुसार त्या विषयांच्या व्याप्ती असतात, त्याच्या काहीतरी मर्यादा असतात आणि त्या व्याप्ती, त्या मर्यादांच्या त्या चौकटीमध्ये राहूनच त्या ठिकाणी आपल्याला किंवा त्या संशोधकाला ते संशोधन पार पाडायचं असतं. कारण संशोधन करण्याला तर अफाट असं दिशा असतात. कुठलाही एखादा विषय घेतला त्याला वेगवेगळी अंग असतात. एवढ्या सगळ्याच अंगांनी जर आपण त्या ठिकाणी ते संशोधन करत जर गेलो तर अजून एका गोष्टीतून दोन गोष्टी, दोन गोष्टीतून चार गोष्टी त्या ठिकाणी जन्माला या येत असतात किंवा त्याला दिशा या मिळत असतात. आणि म्हणून ते संशोधन हे काही वेळेला रटा होण्याची एक संधी जास्त असते की रटा होण्याचं एक त्या ठिकाणी चित्र निर्माण होतं. त्याच्यामुळे त्याला व्याप्ती त्या ठिकाणी टाकणं, त्याचा मर्यादा काय आहेत त्या मर्यादा आखणं आणि मग त्या व्याप्तींच्या अनुषंगाने, त्या मर्यादाच्या अनुषंगाने त्या ठिकाणी ते संशोधन करणं हे फार महत्वाचं असतं. जेणेकरून त्या संशोधनाला निश्चित असं एक प्रकल्प असं किंवा त्या संशोधनाचा अभ्यासपूर्ण रूपांतर, अभ्यासपूर्ण त्याला महत्व त्या ठिकाणी प्राप्त होऊ शकतं आणि त्यानंतरच त्या ठिकाणी मग तो पूर्ण संशोधनाला दिशा मिळत असते आणि ते संशोधन त्या ठिकाणी परिपूर्ण होत असतं. संशोधन करता जो काही असतो त्या संशोधनकर्त्याला ते संशोधन त्या ठिकाणी ज्या वेळेला तो करतो ते संशोधन करत असताना बारीक सारीक फार फार गोष्टींचा त्या ठिकाणी लक्षात ठेवावं लागतं. काही गोष्टींचे नियम त्या ठिकाणी त्याला पाळावे लागतात. पहिली गोष्ट म्हणजेच काय की त्याच्या संशोधनाप्रती त्याची निष्ठा पाहिजे, की मी जे काही काम करतो आहे ते प्रामाणिकपणे ते काम करतो आहे का नाही, किंवा ते प्रामाणिकपणे ते काम व्हायला पाहिजे. आपण हल्ली बघतो की या संशोधन क्षेत्रामध्ये देखील बरंच वाङ्मय चौर्य ज्याला आपण म्हणूया तर तशा प्रकारच्या घटना या ठिकाणी घडत असतात किंवा सहसा आता कोणीही लवकर एकदम ते आपल्याला व्हायला पाहिजे अशा अनुषंगाने कुठून तरी जे काही समाज माध्यम आहेत त्याच्यातून जे काही सगळं रेडीमेड जे काही सगळं साहित्य आहे, काही विषयांचे मुद्दे आहेत, काही विषयांचा पूर्ण संदर्भ आहे, ते जसे तसे घेणं, तसं ते वापरणं आणि काहीतरी घाईगर्दीमध्ये ते संशोधन पूर्ण करून त्याची मांडणी करणं, अशा प्रकारचे बरेच आता घटना या घडत असतात. तर अशा वेळेला त्या संशोधनाला कुठल्याही प्रकारचं आधार राहत नाही किंवा त्याचं जे काही वस्तुनिष्ठपणे जे काही कार्य आहे त्या कार्याला त्याचं ते महत्व राहत नाही. आणि म्हणून जो कोणी संशोधक असेल त्या संशोधकाला सर्वप्रथम काही गोष्टींचे नियम देखील त्या ठिकाणी पाळावे लागत असतात. ते म्हणजेच कोणते कोणते की संशोधनाप्रती त्याची निष्ठा पाहिजे, की मी जे काही करतो आहे ते अतिशय प्रामाणिकपणे ते काम व्हायला पाहिजे. जसं एखादा लहान मुलगा खेळत असतो त्यावेळेला तो खेळत असताना पूर्ण तन मन धन त्याचं त्या खेळामध्ये मग्न गुंग असतो, त्याला कुठल्याही गोष्टीचे भान नसतं. मग त्याचे कपडे खराब होत आहेत, त्याचं शरीर खराब होतोय, कुठेतरी माती लागते आहे वगैरे वगैरे वगैरे. म्हणजे त्याला ती जी काही आवड असते खेळण्यामध्ये तो त्या खेळण्याच्या त्या आपल्या त्या आवडीच्या कामामध्ये तो पूर्ण मग्न त्या ठिकाणी होतो, स्वतःला पूर्ण एकरूप करून घेत असतो. त्याचप्रमाणे ते संशोधन करत असताना संशोधकाने त्या कार्यामध्ये पूर्ण मग्न करून घेतलं पाहिजे. आणि ते काम अतिशय प्रामाणिकपणे एकनिष्ठपणे त्या ठिकाणी घेतलं गेलं पाहिजे, केलं गेलं पाहिजे. तसंच पुढचं वस्तुनिष्ठ ज्या वेळेला ती परिस्थिती संशोधक ज्या वेळेला हाताळतो, तर ते हाताळत असताना जागरूकता ही संशोधकामध्ये असणं ही फार आवश्यक आहे. त्यासाठी मग त्याला जे काही तो विषय त्याने निवडलेला आहे त्या विषयाच्या ज्ञानाबरोबरच जे काही संशोधन आहे त्या संशोधनाची इतर जे काही विषयाच्या अनुषंगाने जे काही बाकीच्या गोष्टी असतील, बाकीचे सगळे पुस्तकं असतील, संदर्भ असेल किंवा अजून काही साहित्य असेल, त्याची देखील त्याला रुची असावी, त्याची देखील त्याला आवड असली पाहिजे. त्याच वेळेला त्या संशोधनाला तो अजून चांगलं रूप कसं देऊ शकेल हा एक प्रयत्न तो करू शकतो. पुढचं की एक तो संशोधक संशोधन करत असताना त्याचं जे काही मनोधैर्य आहे, त्याचा जो काही आत्मविश्वास आहे, तो आत्मविश्वास त्या संशोधकामध्ये अतिशय प्रचंड प्रमाणामध्ये असणं आवश्यक आहे. कारण की ज्या वेळेला तो संशोधन करतो, रिसर्च करतो, बऱ्याच वेळेला अनेक समस्यांना सामोरं जावं लागत असतं. एकच गोष्ट परत परत करावी लागत असते. पहिल्यांदा त्याला त्याच्यामध्ये काही यश येत नाही, दुसऱ्यांदा यश येत नाही, त्यासाठी अनेक प्रयत्न करावे लागत असतात. बऱ्याचदा अपयश येण्याची शक्यता असते आणि बऱ्याचदा अपयश आल्यानंतर कुठंतरी त्याचा आत्मविश्वास हा डगमगू शकतो, कुठंतरी त्याचं धैर्य हे खचू शकतं आणि म्हणून त्या गोष्टीची तयारी संशोधकाने अगोदरच बांधून घ्यायला पाहिजे की जेणेकरून आपल्याला अथक प्रयत्न करावे लागतील. प्रयोग हा एका प्रक्रियेमध्ये साध्य होणारा हा भाग नाहीये, तर अनेक वेळेला प्रयत्न करून त्या ठिकाणी बऱ्याच वेळेला त्याच्यामध्ये अपयश येऊन कुठेतरी शेवटी यश येत असतं. तर कुठल्याही क्षणाला असं कंटाळा न करता, कुठल्याही वेळेला असं निराश न होता सातत्याने आणि चिकाटीने त्या संशोधकाने काम करत राहायला पाहिजे आणि तो चिकाटीने काम करण्याचा गुण त्या ठिकाणी त्या संशोधकामध्ये असणं हे फार आवश्यक आहे. पुढचं की कोणत्याही ठिसूळ किंवा कोणताही कुचकामी पुरावा, कुठलंही संदर्भ नसलेलं विधान, मत संशोधकाने संशोधनामध्ये वापरता कामा नये. जे काही तो मांडणी करत असतो, जे काही विधान तो त्या ठिकाणी, जे काही वाक्य त्या ठिकाणी तो मांडत असतो, त्याला कुठला ना कुठला तरी आधार त्या संशोधनामध्ये देणं हे गरजेचं असतं. कारण संशोधनामध्ये त्या संशोधकाला आपली वैयक्तिक मतं मांडण्याचा त्या ठिकाणी अधिकार नसतो. जे काही त्याला मांडणी करायची आहे तो त्या असलेल्या साहित्याचा, असलेल्या सगळ्या संदर्भाचा आधारे तो नवीन काहीतरी त्या ठिकाणी मांडणी करत आहे आणि म्हणून ते संदर्भ देत असताना किंवा ते वाक्य वापरत असताना ते वाक्याचा कुठल्या ठिकाणी, कोणत्या पुस्तकामध्ये, कोणत्या ग्रंथामध्ये, कोणत्या लेखकाचा, कोणत्या वर्षाचं प्रकाशन, अशा प्रकारे सगळ्या गोष्टींचा संदर्भ, मग ते पेपरातलं कात्रण असेल, एखाद्या मासिक मधलं एखादा संदर्भ असेल, एखाद्या ग्रंथाचा असेल, कुठलाही असू द्या, पण त्या ठिकाणी तो संदर्भानिश्चित त्या ठिकाणी ते वाक्य, ते मत, त्याची मांडणी संशोधकाने त्या संशोधनामध्ये करणं हे आवश्यक असतं. प्रामाणिकपणा, मी मगाशी सांगितलं की प्रामाणिकपणा हेच संशोधकामध्ये असणं आवश्यक आहे आणि प्रामाणिकतेबरोबरच जे काही संशोधन करणार आहे त्या संशोधनातून जे काही निष्कर्ष काढण्याची जी काही क्षमता आहे ती त्या संशोधकामध्ये असली पाहिजे. संशोधन करत असताना शेवटी पूर्ण जेव्हा कार्य पार पडेल त्याच्या शेवटी जे काही ते निष्कर्ष मांडणी होते, तर त्यावेळेला प्रामाणिकपणा मी मगाशी सांगितलं की प्रामाणिकपणाचं असणं आवश्यकच आहे. बिलकुल त्याच्यामध्ये निष्ठा त्या कामामध्ये असणे ही आवश्यकच आहे आणि त्यावेळेला ते निष्कर्ष ज्या वेळेला तो संशोधक मांडतो, पूर्ण संशोधन पूर्ण पातळी त्या ठिकाणी टप्प्याटप्प्याने पूर्ण झाल्यानंतर, तर त्यावेळेला ती निष्कर्ष काढण्याची जी काही क्षमता आहे ती संशोधकामध्ये असली पाहिजे. आणि अशा या सगळ्या गोष्टींनी जर तो संशोधक त्या संशोधनाला जर सामोरं जर गेला तर निश्चितच त्या ठिकाणी त्याचं संशोधन हे अतिशय उत्तमरित्या त्या ठिकाणी पार पडू शकतं. याच्याबद्दल कुठलंही त्या ठिकाणी दुमत नाही. अशा प्रकारे थोडक्यात जे काही तुम्हाला बोलण्याची संधी दिलेली होती आयोजकांनी, त्याबद्दल मी आयोजकांचे खास करून माननीय प्राचार्य पवार साहेबांचे या ठिकाणी मी धन्यवाद देतो आणि माझं 2 शब्द थांबवतो. धन्यवाद. Hello.
Yes, sir. Thank you, sir, for such a great session.
Thank you, sir.
I would like to invite Amit Dhankani sir to continue the session. Amit sir.
Okay. Thank you, Mr. Divakar Patil. Thank you, Dr. Tushar Patel, for explaining the concept of research in our local Marathi language. It will help our students to understand the concept of research in the most easiest way. Thank you, sir. Thank you very much.
धन्यवाद सर.
One notice for participants: the next session will resume at 1:30 p.m. The separate link will be provided for the poster presentation competition. The link will be provided in your WhatsApp group. Now we declare that the session is over.
झालं का सर ते?